Hello, everybody. Welcome back. We've got a near final session. We've got two speakers for you this afternoon, and then there'll be a Q&A session later on at 3:35 BST. So the first speaker today is Thor Rowlands from Nelson Labs. He's the Director of Toxicology there and another member of ISO TC 194. And I'm very pleased to welcome him to talk to us about feedback he's had from the FDA on 510K submissions, which I think is an interesting subject for many people. It's always difficult to work out exactly what the FDA is going to think about any particular subject, and I'm sure Thor has a better idea than most of us. So Thor, thank you very much.
Thank you, Jeremy, and I sure appreciate everyone taking some time today to be with us and talk about this great topic. And I agree with Jeremy. It's the FDA has gotten much better with a lot of their feedback and their approach to try to make things more uniform. But there's still some areas that we've had some issues with routinely with responses for the biocompatibility feedback. And so today we're going to, in the 20, 25 minutes that we have, we're going to touch on some of these topics from FDA feedback and really try to understand why the FDA is kind of asking these questions and how we can better kind of prepare our submissions to be able to predict these type of feedback, make sure we have more of a smooth submission with the FDA. So first off, why now? So why are we having, historically the FDA has kind of gone up and down with how easy it is to submit samples. Sometimes they've been a little easier to get through, other times they've been very difficult. So what's happening right now with what we're seeing with feedback? And it has to do kind of with two main things that changed in biocompatibility, which I know we've talked much about in this conference, which comes from we're changing a lot to how we assess biocomp away from these checkbox mentality of just doing animal testing and more into a risk-based approach. So if you look at the new FDA guidance document, if you look at the ISO 10993-1, these types of approaches to biocompatibility are kind of forcing the FDA to look at each assessment individually instead of that checkbox mentality. So if you remember, that the past approach, I've been at Nelson Labs for 18 years. The whole time I've been in biocompatibility, I've been part of the TC 194 for 10 years, over 10 years now. And so I've seen the past approach, when we had devices that were made from stainless steels and titaniums and polypropylenes. A lot of times we would just, historically, would pull up this chart. This is actually the guidance document from 1995 is when it was put in place by the FDA. And the FDA kind of just checkboxed this. So what the approach was is that you would go through, right, and you would say, okay, my device contacts the body like this. It's for this length of time. Check, check, check. You do those tests and you submit it to the FDA. So this was the past approach that we saw with the FDA for years. Now with the new ISO 10993, when you start looking at the importance of material evaluation and chemical evaluation on the risk-based approach to then evaluate what testing is needed, it's turned much more into, well, what it should be, which is a risk assessment. It's turned much more into trying to understand the materials you're making, that your device is being made out of, the processing that's going into your devices, and then how they're used on the patient. And you take all these inputs and research and toxicological evaluations, and you look at those individual endpoints for biocompatibility to understand if they're needed to mitigate any risks. So for example, if you're looking at systemic toxicity as a biological endpoint that needs to be assessed, do you need to do animal tests, or can you address those risks through chemistry, or do you have sufficient information from materials and processing to be able to feel good about the risks of the systemic toxicity from your device without doing those tests? So this has gone, even though this is the correct approach, in my opinion, to assess medical devices, and I love the fact that we're moving more towards that, especially since I have a bias against some of these animal tests. I don't feel they're as sensitive as we feel they are. But the problem with that type of approach, it does take expertise to be able to put all those inputs together and come out with the right end. And so when you're not putting together the right story, or you're not putting together the right inputs, then submitting to the FDA or really any regulatory agency can have some problems. So what we're going to talk on today is kind of how to look at some of these inputs based on what the FDA is asking going forward. So with this, like I talked about, you do need some expertise. So you need toxicologists, you need chemists, you need biocompatibility experts. These work together in order to really help evaluate that. And you need to put all those inputs together into something that we call BEPs, biological evaluation plans. And my colleague Sophie did a great job earlier in this conference, kind of explaining how we at Nelson Labs look at BEPs. But obviously, there's lots of people who put together nowadays these risk assessments, which we call biological evaluation plans. And the other part of this feedback we're getting from the FDA that we'll talk about in a minute comes from the new extractables and leachables. The new 10993-18, which Ted Heisey had a huge part of, is a great standard. It really is a great standard to help show how chemistry, the power of identifying and quantifying compounds from a medical device, can be used to assess the safety of that medical device. But the issue is this chemistry, this extractable leachable, is not as standard as a sensitization test, or even as an acute systemic or subacute subchronic toxicity test. There's differences on how you can set up an extractable leachable test, and there's differences on the results that are coming from these tests. So this makes it a little bit more ambiguous when you submit to a regulatory agency exactly the quality that you're getting from these tests. So this is just showing you that this is the FDA's approach. So this is the paragraph from their guidance document that helps you understand how to write a BEP, basically. So you look at the materials, you look at the processing, you look how it's used, and then you develop a plan to address those risks. So really, it's the step that the FDA wants you to take. Okay, so when we start getting feedback from the FDA, we start looking at some of these things that we're getting most of the feedback from, and trying to identify those. So that what we're seeing from it is we need to be able to get better identification from the chemistry run, so we need to have scientific detail on identification and what should be reported. Then we're also getting some unpublished kind of changes to expectations from the FDA that we're getting on these feedback. So we're getting more and more guidance on exactly what extraction solvents or extraction parameters we should be using for chemistry specifically. So we're looking at solvents, we're looking at the difference between exaggerated and exhaustive conditions. The sample preparation details are important, method solubility, and then we're even seeing from the FDA themselves, some of the FDA kind of talking about CMRs, even though that's in the new MDR and focused more in Europe. We're starting to see the FDA look at specific CMRs, carcinogens, mutagens, and reproductive toxins, to make sure that we're addressing them based on the materials that we're using and so forth. So to get a little bit more specific on the feedback we're getting, we're going to go into actual text from this feedback here about chemistry and the identification process. So the first one that we're going to talk a little bit about is about this identification of compounds. And as when you're dealing with toxicology, it's almost, as wonderful as toxicologists are, I've never met one that's really been able to assess a compound without knowing what it is and not knowing how much of it's there. That's kind of two of the most important parts of a toxicological evaluation, is identifying and quantifying. And so you can make assumptions sometimes if you have semi-quantitative amounts and maybe structure analysis, but reality, the better the identification, the better the quantification, the better the tox assessment. So these kind of feedbacks we're getting from the FDA right here is specifically generated towards identification. So they're asking you to give them the actual details of how you identify those compounds. So they're asking about the library, the spectral reference library you're using, the match score, the algorithms. Basically, they want you to spell out exactly how you're identifying these compounds. The next feedback we're getting from the FDA, I wanted to kind of very quickly hit on the importance of identification. So when you start looking at identifying, obviously we use mass spectra to be able to help identify and quantify these compounds. And we use this chromatogram to be able to do that. And you can compare that to public libraries like NIST or Wiley, which is a very common approach. The problem with that, though, if you just use these public libraries, the automatic ID can come up with misidentifications without an internal database with expert review. So we really emphasize that the FDA is looking for you to justify, just not that you ran this list in the NIST library and be able to identify it without even looking at the compound and looking at that match. You really have to take some time to go through it with an expert and your internal database to help justify those matches with the FDA nowadays. So, and that just shows you our type of assessment as far as, especially the non-volatiles. There's not really a great public library for non-volatile compounds. So having an internal database is extra important when you start looking at FDA submissions. So when you're looking at what you need to do for your submission, you need to take extra caution when you're choosing your structure associated with the highest match factor. All the potential identifications should be provided to the FDA and a justification of why you chose the identification that was chosen or the lab did. Any substance with only a partial structure should be designated as an unknown compound for the toxicological risk assessment. This is feedback from the FDA. And then all potential members of that class toxicological evaluation should be identified. Okay, so as far as the things we don't, the other kind of extra caution is needed. These are some of the endpoints that we also recommend, that you don't just choose a structure associated with the highest match factor, like we talked about, and they should all be identified. Sorry, I'm going to walk through these. These are the things we just talked about. So now we're going to go through some extra feedback we've been getting on quantification. So these are some of the FDA responses we're getting on just on the identification of compounds, but also on the quantification of compounds. So these are the word by word, some of the submissions just recently we've gotten back. So when you're looking at quantification, it really comes down to the number of internal standards you're using. And that's one of the main things that the FDA is hitting on right now. And I know that the 10993-18 is starting to, that also talks about internal standards to be able to help with quantification. Remember that obviously an identified compound with an exact quantification is the best type of quantification you can get for a toxicological risk assessment. But in most of the times, what we're looking at is a semi-quantitative amount based off of a comparison to an internal standard, which is or a surrogate. And so the more surrogates we have and the more appropriate surrogates we have, the better the semi-quantification of that compound. And when you're doing a toxicological risk assessment, obviously, if you have even off a factor of 2 or 3 off the amount of that compound, can have an impact on your toxicological evaluation. So the FDA knows that, and they're very cautious in asking for the correct number of surrogates to be used and the type of surrogates to be used. So when you're looking at why this matters, so like I talked about, how we do quantification is we compare the unknown or the compound we're looking at to an internal standard to be able to get us the amount of that compound there. So for those of you that may not run chemistry all the time, to understand this is it's not like CSI or other TV shows where we throw something in a black box, we push a button, and it tells us exactly what it is and exactly how much is there and what Walmart or superstore you bought it from. That's not how chemistry works. What really happens is we have a database of compounds that the lab has identified and quantified in their library. But the vast amount of compounds in the world, there's no way a lab will have every single compound that could possibly be there, every degradant or breakdown of that compound. So what ends up happening is you have to be able to try to estimate those compounds by comparing it to something you do know. So what we end up doing is looking at these peaks, comparing those peaks to our internal standard. But if you have a peak that is somewhere, and then that gives you a range to be able to give you a quantification. So that's the step one, is you look at the surrogate, you try to understand what it looks like compared to what you're trying to identify or quantify in your scan. So here we have an estimated quantification analysis with our surrogates. So you can see where our surrogates would be, our 3 surrogates over the time range. And so what we end up trying to do is look at these other compounds that we have identified in the surrogates. And I just lost my connection to the next slide. So can I have someone progress the slides for me? There, it just reconnected. Oh, it skipped all the way to the bottom. Okay, so basically, thank you for that. So to end on today's presentation, these are the feedback we're mostly getting from the FDA. It's focused mostly around extractable leachables. It's focusing mostly on the quantification of these compounds and also the identification of these compounds. And in order to kind of help smooth out your submission to the FDA, especially for the submissions that require chemistry, it's important to work with whatever lab you're working with to be able to correctly identify and quantify the compounds so that when they submit, that the FDA is happy and you're not having to go back and do additional work with the chemistry. Now, obviously, I only had 25 minutes to do this presentation. That we have tons more feedback with the FDA that we get, just not on chemistry, but on sample preparation, on cytotoxicity. We have other educational resources that you can go to our website to be able to get those if you have questions about other things. And at this time, I'd like to open it up to any questions. I think we have about 6 minutes worth of time for questions.
Right, so unfortunately we haven't had the questions through yet. Can you put them on? Oh, here we go. It's just come in. Yeah, if the supplier provides biocompatibility info, but not on the raw, but on processed material, according to their own recommendations, and if we process the materials at the same conditions, could it be more reliable at chemical characterization?
That's a great question, and honestly, that is something that we take in when we look at our biological evaluation plan. So we get a ton of clients that contact us and say, okay, so I don't think I have to do any biocompatibility because this is made out of whatever material it is. And it's been used on the market forever. My competitor uses the same material, whatever. I might have an FTIR scan to show that it's the exact same material. So why do I have to really test it? And those are questions we put into our biological evaluation plans. We do assess the common use of these materials in that type of application. And you're right that if you can find a supplier that actually has testing done with the same processing conditions, that definitely can be used to help mitigate the risk of a medical device. But you have to be really careful because the processing, in my experience, in my opinion, a lot of times the processing actually impacts biocompatibility more than the materials themselves. It is true that lots of people are getting materials from similar suppliers. We see it all the time. We see the same names of suppliers, the same type of materials. but it's not necessarily the materials that concern me when I do the assessment. It's what you're doing to those materials that end up impacting the biocompatibility of them. And so you really want to make sure you go through and be able to, you can prove that you're processing that, and that's including sterilization and even possibly packaging, because I've seen packaging have an impact in biocompatibility to be able to justify the use of that information. But this is all part of your risk assessment. So you want to be able to understand the risk and the inputs that come in, and then there might be some tests we can help justify with that information. It just really depends on the quality of that information and the type of processing you're doing to that device.
Right, thanks, Thor. There's another question here. To make comparison with the other products' peaks in the components, it's suggested that we use the predicate device in the case of a 510K.
No, so, well, most of the time, that's not what we recommend. That is something you could do. You could run a predicate device alongside yours, and that could give you valuable information to help you understand how similar your peaks, your compounds are to a predicate device. But in the reality, unless you actually are making the predicate device yourself, the chances of those scans to be even close to the same is very it's a long shot. So in my opinion, it's better suited that you just run your chemistry on your device, and then you compare it with the internal peaks, like we talked about. There's internal standards to be able to help quantify the amounts. The identification comes from the libraries, and it's important to actually have a laboratory that has the library that can help you identify those compounds. Because the worst thing that can happen in a chemistry test is when you get a bunch of unknown compounds above a toxicological threshold, and then as a toxicologist, you're concerned about those compounds, and how do you make a risk assessment on compounds you don't know what they are, and they're at high levels to make you concerned. So that's really the key to chemistry, is being able to, that the predicate, I don't know if really helps, but really to be able to identify and quantify the compounds is where the power lies.
Okay, thanks. We've got a couple of minutes, so probably time for one more question. Is it correct to establish exhaustiveness through a gravimetric method, which is far from the AET for the device, for example, an implantable device?
Yeah, so this is, so we actually did our own, as Nelson Labs, we did our own pre-sub with the FDA on this, because we don't, scientifically, we struggle with the concept of using gravimetric as exhaustive extraction. And the reason why is, one, you're looking at only non-volatile compounds or high molecular weight semi-volatile compounds, but also the sensitivity in those balances are they're balances. So the amount of sensitivity of the detail you can get down is far away from, like you said, any kind of TTC. The truth of the matter is, though, talking with the FDA, they said that you're either going to use a gravimetric or they want you to use exhaustive on all the techniques. So you'd have to use a GCMS, you'd have to use HPLC and ICP-MS and all these different techniques to get gravimetric, which would be much more costly and time consuming. So from the FDA's point of view, at least in our discussion in the pre-sub, they are interpreting the use of gravimetric as a kind of a compromise with the industry. It's less expensive. It gives you a sense of gravimetric. It does give you a sense of that you're getting a majority of something off and knowing when to start your extractions. And remember, from a toxicological standpoint, a lot of these times we're doing like 72-hour, when you do exhaustive, you're doing your chemistry at that time point. And a lot of labs are saying, okay, so that's your amount. So you can do like a 3 or 4 day extraction, and you say that's your amount, and the toxicologist doesn't know if that's coming off in the first 24 hours or the first 48 hours. So a lot of the toxicologists will just take that 3-day amount and consider it a daily dose. So it ends up becoming a very worst case exposure in a lot of times, the way it's ran for the FDA.
Well, thanks. That's right up to time. So I thank you very much, Thor. It's a really useful presentation and questions.